So this is the third and our last soil session for this afternoon. And with us as instructor is Dr. Carl Rosen. Carl is a professor at the University of Minnesota, as well as a department head with the Soil and Water and Climate Department. So he supervises an active research laboratory related to nutrient and management for crop production. And his responsibilities are both in research and extension, relating to plant nutrition and improving fertilizer use efficiency. His primary emphasis is on irrigated crops, as well as fruit and vegetable crops that are grown commercially in the state. And in recent years, he's also focused on water quality issues related to fertilizer use. So he has a lot of experience, and we're going to make use of it a bit today as he talks about biostimulants.
Very good. Well, thanks a lot. And thanks for the invitation to speak at the meeting today. I was asked to talk about biostimulants for crop production. And I have given this talk a number of times throughout the state and even up in Canada. So hopefully this is the first time you've heard it. I've updated it a little bit, but things have changed since I gave it for the first time about 2 years ago. So biostimulants are a topic of interest to many because there's numerous products on the market and being promoted and sold, probably more than have been in the past. And worldwide, it's one of the fastest growing segments of the agricultural industry and often tied to benefits associated with soil health, which is also another popular topic. There's a lot of venture capital going into biostimulants, and I think a lot of that is kind of in search of a silver bullet, so to speak. So what I'd like to do is kind of take a closer look at what biostimulants actually are. And believe it or not, biostimulants were actually written into the Farm Bill in 2018, and I'll get into that. We'll kind of do a definition of what biostimulants are to many people. We'll get into the types of crop biostimulants. There's many different types that are out there. Look at some application methods. What's the research methodology that goes into what the mode of action might be? And then I have two examples, kind of case studies. One with potatoes. I do a lot of work with potatoes, but another one with corn, where I worked with a cooperator on that one. It's an on-farm study. So what are crop biostimulants? Currently, there's no legal definition in the US, and that's why it was part of the 2018 Farm Bill. I would say in the past, it's been a lot more popular. These products have been a lot more popular in Europe than in the US. And over time, the definition is evolving. So I'll just read you what the definition is from the European standpoint. Plant biostimulants contain substances or microorganisms whose function when applied to plants or the rhizosphere is to stimulate natural processes to enhance benefit, nutrient uptake, nutrient efficiency, and tolerance to abiotic stress and crop quality. One other thing that I want to note with this definition, they don't put crop yield in there, so that's not part of this particular definition. Another definition is very simple. Substances or microbes provided in minute quantities that promote plant growth. And they're very specific to say it's not a fertilizer, a pesticide, or a soil conditioner. So this definition, you can get a little gray in there because there are some of these biostimulants do have some effects on pathogens, root pathogens, and so there is that gray area, and I'll talk about that in a little bit. I also want to mention that there have been, even though it's very popular these days, this is not a new topic. There have been a number of products that have been marketed for many years, and as early as 1980, there was a regional committee formed called NCR 103. And it was developed to test claims of biostimulants and other non-conventional products. So this is something that's been around a lot more. There just seems to be a lot more emphasis in it now. And one of the objectives of this particular committee was to test these products, but there are so many products on the market now that it's kind of overwhelming to the committee members. So we really rely on kind of on-farm trials, having growers kind of do some of their own testing as well. But there is a compendium on some of the products that have been tested, and that website is located on this particular slide if you're interested. So let's go back to the 2018 Farm Bill, and the definition in the Farm Bill, the tentative definition, was a plant biostimulant is a substance or microorganism that, when applied to seeds, plants, or the rhizosphere, stimulates natural processes to enhance or benefit nutrient uptake, efficiency, tolerance to abiotic stress, crop quality, and yield. So in this definition, it's very similar to the European one, but they did add yield into it. So the purpose of including it in the Farm Bill was to get a more uniform definition. And to do that, the USDA coordinated a multi-stakeholder work group. It involved federal agencies, some NGOs, some industry participants. And the common goal was to come up with some labeling that states could follow and to kind of say what actually is a biostimulant and what to expect. Unfortunately, that report is not yet completed, at least not to my knowledge. And so there is still some, the definition is still, as I said before, it's still kind of evolving. So with that in mind, let's just talk about what different type of biostimulants there might be on the markets. And I kind of categorize them into two different types. One are what we call biologicals. And the biologicals are kind of organic substances, things like humic substances, amino acids, or other nitrogen compounds. Sometimes there's sugar or molasses that are in there. Chitosands, things like plant extracts or seaweed extract. And I'll go into a little more detail of what these supposedly do or have been reported to do based on the literature. And then the second category of a biostimulant is something live, microbials. These are beneficial microorganisms that are isolated and then added to a mixture. They're either bacteria or fungi, and they have plant promoting properties to them. And I'll get into some examples of those. Now, these products are usually applied either individually or in combination. Sometimes they'll mix a biological with a microbial. They're promoted as natural, and they're usually applied in very small quantities, usually quarts per acre to maybe a couple gallons per acre. So not real high amounts. So you're not really expecting any nutrient value of these. You're expecting more plant growth promoting properties. So let's talk a little bit about humic substances. These have been around for many years, probably some of the older biostimulants that have been out there. And humic substances are simply constituents of organic matter. They're decomposition products of plants, animals, microbes. And the humic substances that are manufactured usually come from either peat or compost or Leonardite, which is kind of a coal-like product. And you get humic substance naturally just with organic matter breakdown, but the humic substances that are sold are usually come from one of those other products that we just talked, peats or Leonardite or compost. The different types of humic substances are characterized by molecular weight. And there's a long process in which to extract these humic substances from these products. I'm not going to go into that, just know that there is quite a bit of research into isolating humic substances from these products. So what are the modes of action that you might expect from a humic substance? And under adequate conditions of nutrients in solution, so yet you don't really have a nutrient deficiency, but you will still, in some cases, get stimulation of root growth, secondary and in length. Sometimes these humic substances will act as a chelate. They'll complex metal cations, so you get more micronutrient availability. They've been shown in some cases to increase membrane permeability and stimulate nutrient uptake, especially phosphorus. Sometimes these are applied not only to the roots, but also as a foliar application, which also have been shown to have indirect effects on root and shoot growth. However, if you go into the literature and look at how these humic substances actually perform, their performance is somewhat inconsistent and will depend on a lot of factors. So this was a meta-analysis done. It was a number of years ago now, but still is valid. And in this analysis, what they found is that the response to humic substances was affected by the source of humic substance, and you got a better response if the humic substance came from compost than from lignite or leonardite. The rate of application made a difference, and it seemed like it didn't matter as much as the crop that it was grown or the growing conditions. Some of these other factors were more important, the rate of application in particular. And what they found was, based on this literature review, that yes, the humic substances did increase growth. When they did increase shoot and root growth, it might be by as much as 15 to 25%. But half the studies failed to increase growth at all, or only by 5% at most. So they concluded that there was a high variation and in risk to farmer, because there is a cost to these if you're going to be using them. And what they also found is that when there was a positive effect, it was usually due to the higher rates that were probably not economically feasible to apply, and particularly with those that are recommended on the products that are being sold. So humic substances may or may not work. It just depends on the rate, but there is something there. It's just whether you're going to be applying it in the right situation and the rate. So another compound that's often used as a biostimulant are amino acids. And amino acids and peptides are just protein breakdown products. So they normally get large proteins, either from various animal products, for example. These will be broken down, and sometimes you have them from plant waste material as well as plant products. And these can be broken down into individual amino acids, which are kind of building blocks of proteins. You can see some of the uses that not just for plants, it's also used for animal feeds, dietary supplements for humans, and also for crop production. And you can see the advantage for animals, because animals cannot use simple forms of nitrogen. And so they need to use either protein or unlike plants that can use simple forms. So animals, there is a benefit to adding some of these for animal feed, but plants can use inorganic nitrogen, nitrate and ammonium, for example. So the effect of adding amino acids to a starter fertilizer, for example, it's probably not due to the nitrogen in there, it's due to some other effect. So the reported role of amino acids for plants is that, again, like humic substances, they might have a chelating effect. So they might increase micronutrient availability, maybe decrease heavy metal toxicity. They have also been shown to be involved with enzyme regulation. There is some antioxidant activity, so they scavenge free radicals, so they might increase tolerance to stress. And then there might be an indirect effect. So if you're adding these amino acids to soils, you might be stimulating microbial biomass and nutrient cycling right near where those amino acids have been added. And usually when they're added, they're usually applied right in furrow, maybe right over the seed, so you get an immediate effect. A lot of the studies that have been done on amino acids are based on greenhouse studies, and they've looked at both soil and foliar application methods. Now, whether these translate to the field, that's another question. So there are a lot of work, and a lot of these biostimulants, a lot of the work is done in the lab or in the greenhouse, and then getting it out to the field is usually something that that's what we need to test and see if there is any efficacy. Another product is chitosands, and chitosands are linear polysaccharides composed of glucosamine, and glucosamine is just different glucose carbohydrate products. They're treated, the chitosands are made by treating shrimp shells, which is chitin, with sodium hydroxide. And there are a lot of uses of chitosands. It's actually in the medical field. It's been used for cancer treatment, dietary supplements, weight loss. It's been used in winemaking and in agriculture. So there seems to be multiple uses. The effects, the modes of action depend on the way it's applied, but usually, again, it's a seed treatment or a foliar application. And there is some biopesticide activity of chitosands. It's actually registered by the EPA as a biopesticide. There is some evidence that chitosands can stimulate growth, increase photosynthesis, and nutrient uptake. The actual mode of action, they've gotten down to kind of the molecular level, and so it affects cell membranes, alters DNA, and activates defense genes. So there is some evidence that chitosands can be beneficial. However, here's another kind of a meta-analysis on chitosands, and I'll just read this, it's kind of interesting. The conditions that enabled chitosan to increase wheat were so influenced by the environmental or cultural practice that in a given year, the chitosan benefits could deviate up and down from an average of 10% yield increase and reflected on the product credibility. But then they went on to say, on the plus side, chitosan can also increase yields and is edible without being allergenic, mutagenic, carcinogenic. So the application of chitosan directly to the plant part to be consumed is not a major concern. So basically, if you eat it, you're not going to die from eating it. So I guess that's a plus. But so, you know, there is a lot of variability when you use biostimulants. You may or may not get a response. So that was chitosands. Let's move on to another popular one, seaweed extracts. And this has been used for a long, long time. Seaweed itself has been used for agriculture for many years as a nutrient source and compost. But when used as a biostimulant, you're using it as much smaller quantities. So basically, you're taking the seaweed, you're treating it in some manner, and you get a liquid brown product of the seaweed. And like many of these other biostimulants, they're also often used as a dietary supplement. So they're useful for humans or animals as well, like amino acids and even the chitosands. And like these other biostimulants, they're applied directly to the soil or to the plant as a foliar application. And usually the amounts that are applied are very, very small, like 1.5 to 2 quarts per acre. So very small quantity. So again, you're not getting a nutrient value out of this. It's more some other stimulation of plant growth. So as I mentioned, it's not a direct nutrient benefit, it's indirect. The rates applied are also thought to be too low for direct hormone effects, but there is some evidence that the foliar application stimulates the production of hormones within the plant, which in turn may affect growth and stress tolerance. So those are some of what I call the biologicals, things that have been applied, and they've been applied for many years. What's really new now is what we call the beneficial microorganisms. And these are more the free living ones. We've known for many years the practice of microbial inoculation has been very beneficial. Things like the rhizobium for legumes for nitrogen fixation. And this has been known for a long time. But what we're really looking at are more of the free living biologicals or microbials. And these are called microbial inoculants. They're sometimes classified as biopesticides or biofertilizers. And this is where that gray area is. Are they being really used as a pesticide, or are they considered a growth stimulant? So that's something to be keep that in mind. And, if you do get a growth stimulation, is it due to the fact that maybe you had an effect on a pathogen or something like that? But this area is the fastest growing segment of the biostimulant industry. And a lot of this has to do with new DNA techniques for analyzing the soil microbiome and the interest in more sustainable practices. So some of the types of microbial inoculants are free-living bacteria and fungi, and also mycorrhizal fungi. I'm going to focus primarily on the bacteria for this one. And basically, these bacteria are isolated from soil, plants, or plant residues. And in many cases, what these companies will do is identify very high-yielding fields, usually of corn, and they'll extract the bacteria right in the rhizosphere of those roots. And since you have a high yielding situation, the idea is that these bacteria that they isolate are associated with good growth. And some of these are actually endophytes. They'll actually get into the plant itself and have different plant growth promoting properties. So examples of beneficial bacteria include these ones that I have listed here. They're free-living nitrogen fixers, so they're not like rhizobium where they depend on symbiotic association with the plant root. These that I have listed here are free-living, so they're in the soil. They might be right outside the rhizosphere of the plant. Some of them still might be endophytes, but they're not really considered to be nitrogen fixers like rhizobium. A couple studies have shown, like for example, azospirillum can supply up to 7 to 12% of the nitrogen for wheat. And these are bacteria that are just in close contact with the roots. Other bacteria are what we call phosphorus solubilizers. And these will produce organic acids, kind of chelating maybe iron and aluminum, and allowing maybe some of the micronutrients to be more available. And they might also solubilize some of the phosphorus in solution or break down organic matter with the phosphatase enzyme, for example. There's different mechanisms that these bacteria might work and provide increased availability of phosphorus. Some of these bacteria also have iron transport chelators. They'll excrete those and make micronutrients more available. Some of the organic acids that I just mentioned are also in that same mode of action. Some produce volatile organic compounds, for example, and these might have growth stimulation effects on plant growth roots, plant root growth, for example, things like auxin production. In this slide right here, it just has this is in a Petri dish actually, where in the middle, on one side, you don't have any of the bacteria growing in a culture. There's just a single root. On the other side, where you have bacteria growing in the same culture, but walled off from the other side, you can see that you have a stimulation of lateral roots growing on with this particular bacteria. So there is evidence that you do get these compounds that have some growth stimulation effect. So sometimes there may be some direct production of plant hormones, things like auxin and gibberellins, or even cytokinins. So it could be either direct or indirect in terms of growth stimulation. So the question is inoculation with bacteria effective? And like a lot of these experiments with the other biological biostimulants, the results have been somewhat inconsistent. Each teaspoon of soil contains over 1 billion bacteria, for example. So the beneficial microbes need to be, if you're adding these, you're only adding a very small amount relative to the microbial population in the soil. And so they need to compete with that microbial community, the native microbial community, and they'll often lose out. Another factor to consider is that if you're using these microbials for nitrogen fixation, these free-living microbials, if you add nitrogen to the soil, that tends to shut down the nitrogen fixation process. So you may be adding them, and yeah, they'll fix nitrogen if you don't add any nitrogen, but most of our crops, the non-legumes, for example, you'll need to add nitrogen, and so there's this kind of internal control with these free-living nitrogen fixers that will occur. Now, there is a company out there that has genetically engineered some of these microbes to still fix nitrogen under high nitrogen conditions, but that's more the exception than the rule on the products that are being sold right now. So now I want to move on to just two studies very briefly where we've looked at some of these products. This is a study with potatoes where we looked at nitrogen fixers and a microbial enhancer concoction. And basically in this study, we had 3 nitrogen rates, a starter only, where we didn't add any of the biologicals or microbials. But then we had reduced nitrogen at 150 pounds per acre with and without the N fixers. And in one, we had N fixers and the microbial enhancers. The others, we just had N fixers. And then we had that was at a reduced rate. And then we had the same treatments at a more recommended rate at 225 pounds per acre. The microbial enhancers in this case were amino acids, chitin, chitosan, and glucosamine. And the nitrogen fixers included the azotobacter and clostridium. These were applied at about 2.5 quarts per acre in furrow, and we also came back with another application after emergence. The results were, I guess, the bottom line, the results were a little inconsistent. We saw a significant effect of nitrogen fixers and microbial enhancers at the reduced rate at 150 pounds in the first year in 2015, but not at the 225 pound rate. And what you have here, the kind of the orangey red line or bar that's the control. The green, that's the reduced rate, and the blue is the recommended rate. We didn't see any effect in 2015 on the recommended rate, but then in 2016, we did see somewhat of an effect at the higher rate with just the nitrogen fixers in that particular year. So again, I think the bottom line here is that it was somewhat inconsistent between the 2 years, and yeah, but it just didn't make it's not as consistent as we would have liked to see in order to make a recommendation. This is another study. This is an on-farm study that was conducted a few years ago, and it was it's more in southern Minnesota, and it was to test a product called Envita. It's a biological product with supposed benefits for fixing nitrogen in the plant. It's a microbial inoculant, and it's a Gluconobacter genus that's being added. So this was originally, it's an endophyte, actually gets into the root. Doesn't get in the cell, but it does get into the root itself. And it's originally isolated from sugarcane. It's applied as in furrow or as a seed treatment, and it fixes nitrogen supposedly quickly. And in this particular study, there were 3 rates, kind of a control, a 40-pound base rate, 40 pounds plus 50 pounds of nitrogen side dress, kind of the mid rate, and then 40 pounds plus 120 pounds side dress. That's at 160 pounds. That's the high end rate. And then the 2 biologicals were with and without Envita. And the Envita was applied at 2.5 gallons per acre. And this was a replicated, it was a very extensive study, replicated 4 times in a randomized complete block design, and quite extensive work here. And this just shows an aerial of how it was. Each of these strips was a different treatment, either with low nitrogen, medium nitrogen, or high nitrogen, with or without the biostimulant. And then again, showing the across the field, you can see the low nitrogen treatments were obvious in all of these, all those strips where there was only 40 pounds of nitrogen. Again, you can see where the high is very green, low, very yellow, and then medium's kind of in between in color. So here are the results. It was quite surprising. Obviously, we got a significant yield response, but the effect of the biostimulant was actually negative in this particular case. And in each case, with and without the biostimulant at each nitrogen rate, the effect, you had lower yields when the biostimulant was added. And this is rather unusual. Usually we see either no effect or maybe an enhancing effect of the biostimulant, but this is the one study where I actually got a negative effect due to the biostimulant. So the study conclusions is that doesn't really support the claim of increasing yields, at least for this particular year. Didn't replace any of the nitrogen requirement for corn. The reasons for the decline in yield with Envita aren't known. It could have been due to the need of the plant to supply carbon. So it is an endophyte, so there is some carbon going into this microbe. So maybe there was some energy spent to support the microbe. It's just speculation. We really don't know. So this is my last slide, or second to last anyway. So to conclude, biostimulants are substances or microbes applied to plants or plant roots in small amounts that promote growth. The mode of action varies, but can be either a direct or indirect effect by stimulating the microbes, improved nutrient availability and uptake. They could act as a growth regulator or a hormone, and in some cases actually protect against diseases. A lot of the effects are found in controlled lab experiments, but you have to be cautious of that because they don't often translate to field effects. Field response are inconsistent, so really there's really no silver bullet with these, and you have to remember that they do come at a cost. You're adding something, there's going to be some expense involved. And some of the positive effects that we find, I think we need to do more experiments than just 1 year at a time. There could be some false treatment effects that we see. But so the evaluation of biostimulants need to be done over many years, multiple years, to really get a good idea of how they might perform. And we need to look at systematic approaches to define when the situations are that a biostimulant may be beneficial. So this is my last slide. I thank you for the attention, and again, thanks for the invitation to be here, and I'd be happy to answer any questions if there are any.